I want to shift the focus a little bit now to regulatory compliance and challenges. It is top of mind for all of those, anyone who uses viral vectors, and there is still quite a bit of uncertainty around the regulatory challenges uh, as they pertain to viral vectors. And so, Dave, one of the things I want to talk to you about first is, regulatory requirements differ in early research and development in academia and in industrial vector production. Um, in your experience, what are some of the differences between academia and industrial vector production? And if you could talk us through a little bit why they matter right now.
Yeah, that's a great question, and really something I have a fair amount of firsthand experience with. I've been parts of companies that have procured assets from various academic institutions and whatnot. This continues to be a significant challenge, I think, especially for smaller companies. The fact is, and I certainly don't mean this to be controversial in any way, but the bar to get a phase 1 IND, for example, across tends to be pretty readily achievable for a lot of folks out there. Whereas the bar to amend that IND to get ready to produce pivotal and what you hope is ultimately commercial material is incredibly high, almost BLA type high. Maybe not quite that level, but certainly it's quite a delta between the amendment and the initial IND. The fact is that academic institutions don't tend to place a ton of emphasis on CMC requirements and QBD type building blocks and things like that. And there are inevitably some pretty significant gaps to work through upon the handover of that program to a sponsor company. And again, with the size and the resources available, those can be, incredibly difficult gaps to fill. And the, agency's expectations aren't really too concerned with, the woe is me kind of lack of maybe backstory and whatnot. There is an expectation that backstory is pieced together, and that can involve things like very laborious comparability efforts and, filling in a lot of different blanks, let's just say, that weren't existing. That can be, that disparity around what, and I don't want to just say this is, I don't want to say this disparagingly towards academic institutions. There are small companies that had really no intention to go much beyond phase 1 and had a game plan and simply are not going to spend a lot of time and effort to put a lot of sort of later stage pieces in place. Those can be, and with regulatory expectations the way they are, those can be tremendous challenges to try and complete those stories.
Sure, and we'll be speaking directly to and about the small and emerging biotech in just a second, and how. So that's a good transition into that. But before we do, I want to check with Curran. From your experience and your professional tenure, both pre and currently at REGENXBIO, what have you found to be some of the differences between academia and industrial, vector production to date?
Well, I think the early programs that maybe came out of academia, they typically used adherent cell culture. They may have used serum in the upstream process, and then probably ultracentrifugation in the purification, which makes a really highly purified product. So from a safety standpoint, there's not really a concern there, but that's a really, really difficult process to move and scale into a more commercial GMP setting. And it's taken us about 5 years to do so. And what that does is it places a really heavy burden on the requirement to show comparability of the product. And sometimes that's not as straightforward for some of the diseases we're trying to address to say what's comparable. And so there's been a lot of back and forth, I think, in a positive way with FDA about comparability, but I don't think we have a clear picture yet as to what changes in CMC should require what level of comparability assessment. And that's an area that I'd like to see us tighten up so that on the innovator side, we have a chance to get clear direction on that and not get caught very late in a program trying to make changes or try to get comparability assessments done that weren't planned out.
Yeah, agreed. The comparability challenge is one that affects our readers. Certainly, we've actually dedicated entire other cell and gene live events directly to that topic. So, hear you 100%. I want to go back to in Dave's most recent response. He mentioned something that I want to make sure we cover, which is to talk to the small and emerging cell and gene therapy biotechs when working with the FDA to optimize manufacturing processes to meet the increasing demand for high quality viral vectors. How might they best engage, or what might be the best practices for them to engage with the FDA on this? Dave, what are your thoughts on that?
Yeah, it can be tough. It can be tough for smaller companies really to get the ear, if you will, of the agency and whatnot. I mean, that said, you've got to try. You've got to certainly try and engage as early as you can. We've all heard about the benefits of early engagement, early and often, I would say. It's not the agency's obligation necessarily to meet you halfway. I think that they have been, with the emphasis on advanced therapies in general, I think they have been, maybe a little bit more open and willing to dialogue in a less formal basis, if you will, than in the, historical biotech, or certainly in my experience. One other way that's maybe a little bit creative and not available to everyone, but certainly can be useful, is to try and interact or participate in various consortia and whatnot where the FDA may have a presence. The PDA, for example, is one of those groups that has had past and even in some cases current participation that can be incredibly useful, and it can also help you shape things like technical reports that ultimately can be woven into FDA guidance. I mean, the FDA does use things like PDA technical reports as they're shaping their own guidance and whatnot. So that type of kind of out of the box interaction may be helpful as well.